Formylation, Wikipedia audio. In biochemistry, the addition of a formyl functional group is termed formylation. A formyl functional group consists of a carbonyl bonded to hydrogen. When attached to an R group, a formyl group is called an aldehyde. Formylation has been identified in several critical biological processes. Methionine was first discovered to be formulated in E. coli by Marker and Sanger in 1964 and was later identified to be involved in the initiation of protein synthesis in bacteria and organelles. The formation of N-formylmethionine is catalyzed by the enzyme methionylternamate transformylase. Additionally, two formylation reactions occur in the de novo biosynthesis of purines. These reactions are catalyzed by the enzymes glycinamide ribonucleotide transformylase and 5-aminoimidazole 4-carboxyamide riboside transformylase. More recently, formylation has been discovered to be a histone modification, which may modulate gene expression. Formylation refers to any chemical processes in which a compound is functionalized with a formyl group. In organic chemistry, the term is most commonly used with regards to aromatic compounds. In biochemistry, the reaction is catalyzed by enzymes such as formyl transferases. General formylation reaction. Formylation of methanofuran initiates the methanogenesis cycle. The formyl group is derived from carbon dioxide and is converted to methane. In bacteria and organelles, the initiation of protein synthesis is signaled by the formation of formylmethionylternate. This reaction is dependent on 10-formyl tetrahydrofolate, and the enzyme methionylternate formyl transferase. This reaction is not used by eukaryotes or archaea, as the presence of tRNA-F-met in non-bacterial cells is dubbed as intrusive material and quickly eliminated. After its production, tRNA-F-met is delivered to the 30s subunit of the ribosome in order to start protein synthesis. F-met possesses the same codon sequence as methionine. However, F-met is only used for the initiation of protein synthesis and is thus found only at the N-terminus of the protein. Methionine is used during the rest translation. In E. coli, tRNA-F-met is specifically recognized by initiation factor F2, as the formyl group blocks peptide bond formation at the N-terminus of methionine. Once protein synthesis is accomplished, the formyl group on methionine can be removed by peptide deformylase. The methionine residue can be further removed by the enzyme methionine aminopeptidase. Two formylation reactions are required in the 11-step de novo synthesis of inosine monophosphate, the precursor of the purine ribonucleotides AMP and GMP. Glycinamide ribonucleotide transformylase catalyzes the formylation of GAR to formyl glycinamidine riboside in the fourth reaction of the pathway. In the penultimate step of de novo purine biosynthesis, 5-aminoimidazole 4-carboxyamide riboside is formulated to 5-formaminoimidazole 4-carboxamide riboside by ICAR transformylase. Per-N-GAR transformylase is found in eukaryotes and prokaryotes. However, a second GAR transformylase, PET-GAR transformylase, has been identified in E. coli. While the two enzymes have no sequence conservation and require different formyl donors, the specific activity and Km for GAR are the same in both PET and Per-N-GAR transformylase. Per-N-GAR transformylase 1-CDE uses the coenzyme N10-formyl tetrahydrofolate as a formyl donor to formulate the alpha amino group of GAR. In eukaryotes, Per-N-GAR transformylase is part of a large multifunctional protein, but is found as a single protein in prokaryotes. The formylation reaction is proposed to occur through a direct transfer reaction in which the amine group of Gar nucleophilically attacks N10-formyl THF, creating a tetrahedral intermediate. As the alpha amino group of Gar is relatively reactive, deprotonation of the nucleophile is proposed to occur by solvent. In the active site, ASN 106, His 108, and ASP 144 are positioned to assist with formyl transfer. However, mutagenesis studies have indicated that these residues are not individually essential for catalysis, as only mutations of two or more residues inhibit the enzyme. Based on the structure, the negatively charged ASP144 is believed to increase the pKa of HIS108, allowing the protonate imidazoline group of HIS108 to enhances the electrophilicity of the N10-formyl THF formyl group. Additionally, HIS108 and ASN106 are believed to stabilize the oxyanion formed in the transition state. PTGAR transformylase requires formate as the formyl donor and ADP for catalysis. It has been estimated that PTGAR transformylase carries out 14 to 50% of GAR formylations in E. coli. The enzyme is a member of the ADP-GRASP superfamily of proteins. A sequential mechanism has been proposed for PTGAR transformylase in which a short-lived formyl phosphate intermediate is proposed to first form. This formyl phosphate intermediate then undergoes nucleophilic attack by the GAR amine for transfer of the formyl group. A formyl phosphate intermediate has been detected in mutagenesis experiments, in which the mutant PTGAR transformylase had a weak affinity for formate. Incubating PTGAR transformylase with formyl phosphate, ADP, and GAR, yields both ADP and FGAR. This further indicating that formyl phosphate may be an intermediate, as it is kinetically and chemically competent to carry out the formylation reaction in the enzyme. A enzyme phosphate intermediate preceding the formyl phosphate intermediate has also been proposed to form based on positional isotope exchange studies. However, structural data indicates that the formate may be positioned for a direct attack on the gamma phosphate of ADP in the enzyme's active site to form the formyl phosphate intermediate. Formylation reactions in biology. ICAR transformylase requires the coenzyme N10-formyl tetrahydrofolate as the formyl donor for the formylation of ICAR to FAICAR. However, ICAR transformylase and GAR transformylase do not share a high sequence similarity or structural homology. The amine on ICAR is much less nucleophilic than its counterpart on GAR due to delocalization of electrons in ICAR through conjugation. Therefore, the N5 nucleophile of ICAR must be activated for the formylation reaction to occur. Histidine 268 and lysine 267 have been found to be essential for catalysis and are conserved in all ICAR transformylase. Histidine 268 is involved in deprotonation of the N5 nucleophile of ICAR, whereas lysine 267 is proposed to stabilize the tetrahedral intermediate. Epsilon formylation is one of many post-translational modifications that occur on histone proteins, which been shown to modulate chromatin conformations and gene activation. Formylation has been identified on the N-epsilon of lysine residues in histones and proteins. This modification has been observed in linker histones and high-mobility group proteins. It is highly abundant and it is believed to have a role in the epigenetics of chromatin function. Lysines that are formulated have been shown to play a role in DNA binding. Additionally, formylation has been detected on histone lysines that are also known to be acetylated and methylated. Thus, formylation may block other post-translational modifications. Formylation is detected most frequently on 19 different modification sites on histone H1. The genetic expression of the cell is highly disrupted by formylation, which may cause diseases such as cancer. The development of these modifications may be due to oxidative stress. In histone proteins, lysine is typically modified by histone acetyltransferases and histone deacetylases. The acetylation of lysine is fundamental to the regulation and expression of certain genes. Oxidative stress creates a significantly different environment in which acetyllysine can be quickly outcompeted by the formation of formyl-lysine due to the high reactivity of formyl phosphate species. This situation is currently believed to be caused by oxidative DNA damage. A mechanism for the formation of formyl phosphate has been proposed, which it is highly dependent on oxidatively damaged DNA and mainly driven by radical chemistry within the cell. The formyl phosphate produced can then be used to formulate lysine. Oxidative stress is believed to play a role in the availability of lysine residues in the surface of proteins and the possibility of being formulated. Inhibition of enzymes involved in purine biosynthesis has been exploited as a potential drug target for chemotherapy. Cancer cells require high concentrations of purines to facilitate division and tend to rely on de novo synthesis rather than the nucleotide salvage pathway. Several folate-based inhibitors have been developed to inhibit formylation reactions by GAR transformylase and ICAR transformylase. The first GAR transformylase inhibitor lamotrexol 5,10-diazetetrahydrofolate was developed in the 1980s through a collaboration between Eli Lilly and academic laboratories. Methanogenesis. Formylation in protein synthesis. Although similar in structure to N10-formyl-THF, lamotrexol is incapable of carrying out one-carbon transfer reactions. Additionally, several GAR-based inhibitors of GAR transformylase have also been synthesized. Development of folate-based inhibitors have been found to be particularly challenging as the inhibitors also downregulate the enzyme folate-polyglutamate synthase, which adds additional gamma-glutamates to monoglutamate folates and antifolates after entering the cell for increased enzyme affinity. This increased affinity can lead to antifolate resistance. Formylation reactions in purine biosynthesis. GAR transformylase. Per-N GAR transformylase. Mechanism. PIT-GAR transformylase. Leigh syndrome is a neurodegenerative disorder that has been linked to a defect in an enzymatic formylation reaction. Leigh syndrome is typically associated with defects in oxidative phosphorylation, which occurs in the mitochondria. Exome sequencing has been used to identify a mutation in the gene coding for mitochondrial methionyltRNA formyltransferase in patients with Leigh syndrome. The C.626CT mutation identified in MTF-MT yielding symptoms of Leigh syndrome is believed to alter exon splicing, leading to a frameshift mutation and a premature stop codon. Individuals with the MTF-MT C.626CT mutation were found to have reduced FMT levels and changes in the formylation level of mitochondrially translated COX-1. This link provides evidence for the necessity of formulated methionine in initiation of expression for certain mitochondrial genes. Mechanism 2. ICAR transformylase. Mechanism 3. Formylation in histone proteins. Formylation in medicine. Formylation reactions as a drug target. Leigh syndrome.